friends, my name is Debaditya and welcome to my channel Biologically. In this channel, we will discuss about the important topics of biology and associated fields in a logical way. Now, you guys may have a doubt that why I have created a channel on biology, knowing that there are several thousands of channels already working on it. The reason is, you guys also know that those channels cover some part of the topic, or they just read the slide, or they have a platform to promote where you have to pay and learn, which also they never did. So these things will never build your concept properly. For this reason, I felt the urge to make videos and help you as much as I can. Now, I am making a series of videos on prokaryotic DNA replication. In this series, there will be 4 parts. In part 1, that is in this video, we will discuss about the replication initiation. In part 2, we will learn about replication elongation. In part 3, there will be replication termination. And in the last part, that is the most important part, we will discuss about the previous year's questions of various entrance exams like NEET, JAM, DBT-JRF, NET, etc. so that you can relate with the questions asked in these exams. So without wasting any time, let's start our today's video. First of all friends, in this topic, you have to know that what is DNA replication. So simply I can say that it is the biological process of producing 2 identical copies of DNA from an original DNA molecule, and it occurs in every living cell with a prominent nucleus. Now, you may have a doubt that is the process of DNA replication same in both prokaryotes and eukaryote, and if not, what are those? My answer will be no. The steps will be the same, but the process in prokaryotes and eukaryote are not exactly the same. Because you also can easily understand that the nucleus in prokaryote is not completely developed as of eukaryote. Second, the prokaryotic DNA is circular, but the eukaryotic DNA is linear. Third, you can say that replication takes place in cytoplasm in case of prokaryote, but in eukaryote it occurs in nucleus. The DNA polymerase enzymes are also different. So that is why the mechanism must be different. And the prokaryotic replication is called theta replication. The eukaryotic replication is called linear DNA replication. And another type of replication is present, that is rolling circle replication, which happens in plasmid DNA and the genome of bacteriophage, etc. So these things, the types of DNA replication, I will discuss in a later video. Now, what is the purpose of DNA replication? Simply to produce 2 identical copies of daughter DNA, which is essential for cell division, because each cell will receive a copy of DNA. Now, is DNA replicating infinitely? So my answer will be no. DNA can't replicate infinitely in a normal cell because the telomere of chromosome is shortening by some extent after each round of replication. But in cancer cells, the DNA has infinite replicative potential. I will discuss it elaborately in the 3rd video of replication, that is the termination of replication. Now, what is the difference between chromosome and DNA? So here you can see there is a cell, and within the cell there is a nucleus, and within the nucleus the chromosome lies. So what is chromosome and what is the relation between chromosome and DNA? So the chromosome is nothing but a condensed and coiled DNA molecule packaged with histone and chaperone protein. Now what is gene? Genes are specific sequence of nucleotide within the DNA that can specify a functional polypeptide. And within the nucleotide sequence, there are nucleotide bases called adenine, guanine, thymine, cytosine, and in case of RNA, uracil will be present instead of thymine. You guys also know that. So let's proceed to general features of DNA replication. Here you can see that first the two complementary strands of DNA unwinds and the replication fork forms. Now the replication proceeds in 5' to 3' direction and the process is semi-discontinuous. So what is semi-discontinuous? So here semi-discontinuous means one strand is synthesized continuously in 5' to 3' direction and its direction is equals to the direction of fork movement. So this strand is called leading strand and the other strand is synthesized discontinuously and it is away from the movement of replication fork. It is called lagging strand. And DNA replication will be bidirectional in case of prokaryote, but in eukaryote replication fork proceeds unidirectionally. DNA replication is semi-conservative. What is semi-conservative means? The newly synthesized daughter strand contain one parental strand and one daughter strand. And replication begins in the origin of replication or oriC region and the elongation process occurs by the enzyme DNA polymerase. Here you can see that this is the overview of DNA replication and the replication process originates in the oriC region and it proceeds bidirectionally in case of prokaryote and forming leading strand and a lagging strand. The leading strand is synthesized continuously in 5' to 3' direction and the lagging strand is synthesized discontinuously. And here the enzymes included in this process are DNA pol 3, the main culprit, the DNA pol 1, the DNA ligase, DNA primase, DNA gyrase and DNA helicase. Everywhere you can see that there is something studied. There must be a model organism. So in case of prokaryotic DNA replication, the model organism is Escherichia coli or E. coli. Here is a peak of an dividing E. coli in 0.5 micron electron microscope. And the process starts with the formation of a replication bubble in the origin site or oriC site and it proceeds bidirectionally until it reaches the terminus and then they separate to form two daughter DNA. Now the question is how DNA replication starts. So let's discuss. Here you can see that it starts in the oriC region which is derived from Origin of Chromosomal Replication and it is a unique site that contains 3 types of sequence. What are those 3 types of sequence? First, there are 80 rich 13-mer motif where the melting occurs and in the oriC there are 3 13-mer motifs. Second, the 9-mer motifs where the DnaA protein bind and there are 5 9-mer motifs. Third, the GATC methylation site where the methylation takes place. Now, the protein factors required in initiation are DnaA, DnaB, DnaC, DnaG, SSB or single strand binding protein, DNA gyrase or DNA topoisomerase 2 and DAM methyltransferase. Now, what are their roles in initiation? DnaA protein recognizes the oriC sequence and open the duplex at a specific sequence. DnaB or it is called as helicase, it unwinds the DNA. DnaC, it is termed as helicase loader and it loads the DnaB protein to a single stranded DNA molecule. DNA primase or DnaG synthesizes a RNA primer. SSB protein binds to single stranded DNA to prevent re-annealing DNA Gyrase reveals the topological stress, and DAM methyltransferase methylates the GATC sequence at oriC. Now, what is the actual mechanism of replication initiation? You can see here that first the DnaA binds to the 9' motif of oriC. Then the DIA associates with DnaA bound to the DNA. Then the DIA facilitates the recruitment of an additional DNA proteins. Then binding of DnaA protein facilitates strand separation in AT-rich 13' motif. So how can this DnaA protein facilitates unwinding? The reason is DnaA include two DNA binding domains. One domain binds with the 9' motif and the other domain bind with the 13' motif only when it is bind to the ATP at the oriC. Now the DnaB hexamer is introduced to each strand. Then the DnaC or helicase loader loads the DnaB to each single strand and the helicase become activated. Now the DnaB moves on the lagging strand template in 5' to 3' direction, unwinding the duplex at 5' to 3' polarity. And the chemical energy required for this directional movement is provided by ATP. Then the separated strands are inhibited from reannealing by SSB or single strand binding protein, which bind cooperatively in a sequence independent manner. Now the DnaG is introduced by the DnaB protein and it synthesizes RNA primer in each template strand. Because the DNA polymerase enzyme cannot incorporate DNTPs or deoxyribonucleoside triphosphates without a 3' OH end. Then the RNA primer causes the DnaC or helicase loader to release from DnaB or DNA helicase. Now the DNA replication initiates and a primosome is formed. So what is primosome? Primosome is nothing but the functional complex formed by DnaG and DnaB and sometimes other accessory proteins such as SSB, DnaC, etc. Then after initiation, the elongation occurs by the DNA polymerase enzymes, and which I will discuss in the next video, that is the DNA elongation. Now we can summarize the sequence of events during initiation process. There are 5 events occurring during initiation. First, the binding of DnaA protein to 9' repeats of oriC. Then the loading of DnaB or helicase. Then the DnaB opens the helix and binds the DnaG or primase to form a primosome. Then the synthesis of RNA primer occurs. Now the initiation of DNA polymerization by DNA polymerase begins, or the replication elongation proceeds. Now you can ask me that what is the function of DAM methyltransferase? Here you can see that how important it is in this replication initiation process, because the reinitiation of replication occurs only after the methylation of oriC of newly synthesized daughter strand happens, and DAM methyltransferase, this enzyme, methylates the GATC sites of oriC of newly synthesized strands. So here lies its importance. And another protein, SeqA protein, help to prevent immediate reinitiation of replication by binding on the 5' GATC 3' methylation sites and prevent the immediate reinitiation. So that's all for today. I hope this video will help you a lot. If you guys enjoyed, Please hit the like button and share the video and subscribe to my channel for later videos. And if you guys have any doubt, please let me know in the comment section and make sure you press the bell icon to stay updated. Thank you.